Hi, this is Umar. Here we will discuss about the macrophage signaling pathway and we will also understand about the significance of the macrophage. So the macrophage is the M1 and M2 polarization of macrophage is tightly controlled process and entailing a set of signaling pathway. This is the set of signaling pathway which that is used, and macrophage means the big eater, and their function we will discuss. If so, the macrophage ingest pathogens, cancer cells, microscopic debris, for example the microbacterial microorganism, pathogen, virus, and destroy it also. And it is also engulf the neutrophil for the neutralizing the pathogen after the engulfing the material which that are present in the neutrophil. So recycle the harsh chemical, and enzyme and macrophage also play a large role in controlling in inflammation. Malfunction can impact inflammatory disease, so that is basically is malfunction of the macrophage can cause the disease, because the clearance of the pathogenesis will not occur, because this is the part of innate immune system. So here is a bone which that contain bone marrow and stem cell. Inside the bone marrow produce monocyte. This monocyte, after the infection of the tissue, will go the inflammatory cytokines, the inflammatory mediators, which are also the growth factor and cytokine, which that will be bound with the monocyte. And after this binding with the monocyte inflammatory mediators, to lead proliferation to convert the monocyte into the macrophage. So after this macrophage proliferation and the growth factor receptor basically the bind with the monocyte to proliferate to form a differentiated macrophage. This differentiated macrophage contain toll-like receptor which that recognize the antigen, and on the other pathogen. And here the G couple protein receptor used for the growth factor, and macrophage is here contains scavenger receptor used for the engulfing the lipoprotein for decreasing in during the atherosclerosis. So here is the macrophage 1 and 2 will be formed. So let's begin to understand the microbe adheres to phagocytes and general. Our we will discuss here the macrophage forms pseudopores that uh, and uh, and eventually engulf the particle. After the eventually engulf the particle, we move in, so it is known as a phagocytosis. And this phagocytic vesicle containing antigen is known as phagosome, and lysosome will be bound within it. And in this way, the lysosome contain lysozyme enzyme and other hydrolytic enzyme, and neutralizing the phosphatase and proteinase enzyme. So the a phagocytic vesicle is fused with a lysosome and phagolysosome formation. And this lysosomal enzyme killed and digest this content. And in this way, this is the long process we will not understand in this video, but I had made another video. You can watch that. I will give the link in description below. I hope so. So here is the residual body, and indigestible residue will be exocytosed, and this will be cleared by the another mechanism, and also the another macrophage can also use for neutralizing again and again. So here is the immature macrophage. This immature macrophage will be, I am drawing the bigger picture for understanding the stimulation and activation of the macrophage with different manner and the different pathway we will discuss here. So in this way here is I am separating the different pathway in the macrophage. So here is the bilayer of phospholipid plasma membrane, and here you can see this is the EPO receptor which that bind the EPO and bind with the genus kinase 2 will be recruit and docking the stat 3. After the binding with the stat 3 phosphorylation will be dimerization. So remember about that. this monomer will be dimerized, and after the dimerization will lead to become a phosphorylation with the transcriptional factor bound with the DNA. So remember. So on the other hand, the other receptor is the interleukin 4 and interleukin 13 receiving receptor, which that is the tyrosine kinase receptor both, and will bind the genus 3 kinase and stat 6 to become a transcription factor. So after this binding with the DNA, to transcribe the gene, for transcribing the gene for the proliferation, as well as to produce the cytokines, these STAT3 and STAT6 produce the cytokine which that will recruit the other lymphocytes for combating the pathogen, and neutrophil recruitment, and CD8 and CD4 recruitment, and B cell. So this is the here is the JAK3 and STAT3 pathway is this. While here is the retinol will convert into the retinal, and this retinol dehydrogenase enzyme used to ALDH the basically the convert into the ATRA and retinoic acid. So basically is the you can see this is the uh, retinoic acid will be formed. So this is basically here you can see this will be bind with the retinoid A receptor and retinoid X receptor binding the dimerization, and in this way the transcriptional factor will enhance, and after this enhancing the transcriptional factor will proliferation and differentiation. On the other hand, let's begin to understand the mTOR pathway. So what is this? Mtar pathway is used for the proliferation and cell growth. So that is why the monocyte will become a macrophage. So here is the ligand will bind with the tyrosine kinase receptor. For example, growth factor will stimulate the PIP2 to PIP3, and after the PIP3 will be stimulate phosphoinositide triphosphate to activate the PDK1. So the PDK1 will be activate the AKT pathway. So this is also known as the AKT pathway or mTOR pathway. Will stimulate the mTOR complex, mammalian target of rapamycin. So this is the name. And differentiation and growth will be occur, and in this way the cell autophagy will be inhibit. So the differentiation to produce macrophage 2. So this is the basic activation of the macrophage. So let's begin to understand another the necrotic factor kappa B binding. So after the binding with the necrotic factor kappa B, which that is used for the basically is the macrophage will be killed. Macrophage will kill. But here first of all we will understand about the another ligand which that is used for the proliferation also the macrophage is receptor tyrosine kinase will bind to recruit the ARC, GNK and P38 protein by the MAP kinase. So the kinase enzyme used for the activation by the phosphorylation inactive to activate the MAP kinase is used for it. So the mitogen activated protein kinase, this is known as. So to activate the DNA as a transcription factor to translation to differentiate and proliferation will be occur, and in this way the proliferation is same things you should need to understand, and this is basically the proliferation. Anyhow, in this way this you can see this is the basically the proliferation system, and while arc is very important for the proliferation and cell growth to become a macrophage from the monocyte. So that is basically the chemokines and inflammatory mediators which that will be bind with the monocyte to proliferate. So here is a ligand, which the ligand is the necrotic factor kappa B receptor, is the toll-like receptor 4, and this toll-like receptor 4 is used for the important thing is that used for the pathogen associated molecular pattern will be recognized to stimulate the P65 and P50 protein. This P65 and P50 protein is basically after the recognition to mingle activation also from P65 to SYK gene activation and SYK protein activation to proliferate to stimulate the P50, and this is the binding as a transcription factor with the DNA. After the binding with the transcription factor with the DNA is used for the endocytosed also and, and produce the necrotic factor kappa B, which that is used for the production and the lysis of the cell. So in this way here macrophage will be proliferate and the damage the pathogen associated molecular pattern, and in this way the toll-like receptor is very important for the recognition of the pathogen. While here is, let's begin to understand the mechanism. Here is the infected tissue. This infected tissue will represent the antigen presenting cell MHC class 2 molecule and MHC class 1 molecule. After this MHC class 2 molecule, which that is present, and in this way here is the TMPR is the receptor also recognized and also produce the inflammatory cytokine, will recruit the macrophage. Also the recruitment of the CD4 and CD8 lymphocyte, T lymphocyte, and in this way here is the macrophage also engulf the pathogen, which that is related to the infected cell. So it will clear the infection before the invading pathogen into the cell. So the inflammatory cytokines will be produced after the engulfing and chemokine release, and after this releasing will bind with the CD4 and CD8 T cell. After the binding with the CD8 and CD4 T cell will recruitment, and in this way the recruitment of the CD8 and CD4 via a macrophage toward the basically is the macrophage is the inflammatory cytokine and give the feedback and recognized by the MSC class 2 and MSC class 1 molecule for the killing through CD8 T lymphocyte and recognize the antigen through CD4 T lymphocyte, and in this way produce the antibody by the stimulation of the B cell for B memory cell and B plasma cell. On the other hand, the damaged inflammatory signal will be also recognized, and in this way these all things basically you should need to understand to lead to cell death. Also after the death of the cell will produce the inflammatory mediators also for recruitment of more macrophage for clearing the dead cell, for clearing of the dead cell via a macrophage. So the macrophage play big role for the clearance of the pathogenicity in the human body, and the macrophage are present in the throughout the innate immune system, so that is present in the mucosal layer of the digestive tract, gastrointestinal tract, and other. There are a lot of things which that, for example, the Kappa cell are present in the hepatocyte, and on the other hand there are many more. So here is the some pathogen can invade in macrophage but unable to destroy. Unfortunately, no immune response can damage and kill the macrophage to lead to down regulation of the immune response, lead to disease. There are many disease, infection disease. So I hope you make sense about these things. This was the macrophage signaling pathway. Thanks for watching. Please make sure to subscribe, like, and share.